Hi, my name is Ben Liu, and I'm an 8th grader at Dana Middle School of Arcadia, California. My project title is a homemade, microfabricated, lab-on-a-chip device for urinalysis. With this project, I placed first in the category of animal biology at the LA County Science and Engineering Fair. I also placed honorable mention in the category of biochemistry and molecular biology at the California State Science Fair. I was awarded the title of semifinalist in the 2014 Broadcom Masters National Competition, and I was accepted into the Southern California Junior Academy of Sciences Research Training Program. I had two main objectives during this project. My first objective was to develop an easy and inexpensive method to fabricate microscale devices, with feature sizes as small as 10 microns, at home. My second objective was to design, fabricate, and test a homemade lab-on-a-chip device to separate urine sediment based on particle size differences, and to study their morphology to conduct analysis on patients' urine samples. The problem I am addressing is the need for better methods of urinalysis. Urinalysis is the study of the particles and components of urine. Urine is a common signal for diseases and disorders, and can be used for diagnostics if studied correctly. The properties of urine in patients reflect significantly on the health of their digestive and urinary systems. For example, the discovery of white blood cells in urine are indications of urinary tract infections. Microscopic examination is the most commonly used method for urinalysis. It involves the placement of urine on a glass slide and microscopic examination. The problem with this method of urinalysis is that particles of the urine are randomly distributed across the glass, making urinalysis inaccurate and very prone to human error. As observed in this table, the particles in urine have a large range of sizes. I took advantage of this for my design. As observed in Figure 3, the design of my device was mainly centered around microfilters, which are the main source of my separation. The channel, 2 millimeters wide, 15 millimeters long, and 50 microns deep, consists of 10 microfilters, with opening size ranging from 10 microns to 100 microns. As a urine sample flows through my channel at a flow rate of about 2 millimeters per second, particles are automatically separated by microfilter structures and sorted into microchambers. The purpose is to organize particles based on their size differences for easier and more accurate morphology studies. A large portion of my project was also centered around the fabrication of my device. Normal fabrication of microscale devices involves very labor-intensive, expensive semiconductor bioequipment. So, I wanted to create my own easy and inexpensive method for fabricating microscale devices. This fabrication process started out with designing my device using AutoCAD software. I then sent this design to a local printing shop, and they printed out a transparent mask. And I used this mask to create an SU8 epoxy-based photoresist mold on a silicon wafer by using UV photolithography, using an ultraviolet lamp I bought from Home Depot. Next, I cast this PDMS microchannel using the SU8 mold in an oven at 75 degrees Celsius. I peeled this PDMS off the mold after I was finished. And lastly, I drilled holes into my PDMS microchannel, and I placed this microchannel onto a glass slide. These are images of my fabrication. On the top left corner, you can see my casting of my PDMS mold into my oven. And on the top right corner, you can see the finished process, which is the PDMS sealed onto the SU8 mold. On the bottom left corner, you could see me peeling my PDMS from my SU8 mold. And after I cut this microchannel into a rectangle and I drilled holes into it and I placed it onto a glass slide, I was done with my finished product. I had a very fixed procedure for testing my device. At first, I would load a urine sample through my device using a syringe. I would then move my microchannel under a microscope for imaging and analysis. After testing, I would remove the urine in my device and rinse it. The images are then analyzed for complexity, shape, size, and anatomy, for determination of the health status of patients with these urine samples. Analysis is concluded based on particle morphology. As you can observe in this image, these are pictures of my microfilters, my inlet and outlet holes. I used microscopy to verify that all the features in my filters came out during fabrication. Moving on to my results. My tests with human urine samples demonstrate that my device with pore sizes of 10 to 100 microns was successful in sorting and separating the particles in the urine. Each device consists of 3 channels, each of which has different microfilters for specific types of urine and particle separation. The PDMS microchannel produced many clear images of white blood cells, red blood cells, casts, crystals, and yeast. All of these images were studied to determine their patient's urinary and digestive system overall health status. In conclusion, the objectives I set out for this project were achieved. As demands for more effective detection methods of urinary tract infections and disorders increase, demands for advanced diagnostics devices are also increasing. My science fair project is based on a microfluidic lab-on-a-chip device I invented for urine sediment morphology analysis. Fabricating a lab-on-a-chip microfluidics device at home with features as small as 10 microns sounds impossible because it typically requires very expensive industrial lab equipment. In this project, I successfully conducted the following works, which included developing a simple and low-cost technique to make microdevices at home using household appliances and cheap UV lamps. This allows microfluidic devices for urinalysis to be homemade, low-cost, and reusable for hundreds of tests. Using these fabrication methods, microchannel devices with filter structures of 10 to 100 microns were made for urine sediment morphology analysis. My device successfully separated urine particles based on size differences. Urine can indicate major problems in the digestive system and urinary tract better than any other biological fluids. With my design, I was able to separate and sort particles, followed by morphology analysis, leading to organized, accurate, and effective tests. It is possible to build homemade, cheap microscale devices for effective urinalysis. This is one of the first times such a lab-on-a-chip device was successfully demonstrated for urine sediment particle sorting and morphology analysis. In the future, I plan to develop even more effective microdevices for urinalysis. Although I achieved successful results with microfilters, I hope to use inertial effects and acoustic transducers for separation in the future, because microfilters are not the most effective methods for separation. My device and applications to the urinalysis industry have potential. Therefore, I want to investigate the possibility of presenting my device to industries and scientists for future modifications and possible commercialization.